Reoviruses were first discovered in the 1950s after isolation from human enteric specimens, and they were subsequently documented to infect a wide range of hosts. The term reovirus is an acronym for Respiratory Enteric Orphan Virus, which emphasizes the anatomic site from which these viruses were initially isolated, as well as the fact that infection of humans, although common, is only rarely associated with significant disease. Human infection primarily results in either asymptomatic infection or mild self-limited symptoms such as upper respiratory tract illness and gastroenteritis. In terms of virus characteristics, reoviruses are non-enveloped icosahedral with a double capsid structure. They're about 80 nanometers in diameter. They have pentameric turrets that sit on the outside of the innermost capsid. The outer capsid has a T equals 13 icosahedral symmetry, and the inner capsid has a T equals 2 icosahedral symmetry. The reoviruses have segmented double-stranded RNA genomes. Genome segments number from 9 to 12 depending on the species of virus. Genome reassortment has likely contributed to their wide host range and great success as a family. The innermost capsid layer encloses the double-stranded RNA segments and enzymes for RNA synthesis. Upon infection, transcriptionally active genome segments remain within the cores rather than being released into the cytoplasm. However, newly synthesized messenger RNAs are released into the cytosol through pores at the five-fold axes of symmetry. So these are very unusual viruses. First of all, a double-stranded RNA genome, multiple layers of capsid, although most frequently they have 2 layers. Some species have a 3rd layer of capsid. I threw them in this particular lecture simply because they have the multiploid genome. So as I said, reoviruses have segmented double-stranded RNA genomes, and the genome segments can vary from 9 to 12 in number. So this is a type of virus that you would see reassortment occurring with. For the most part, each segment encodes a single protein. For example, rotavirus and orbivirus genomes have 11 segments encoding just 12 proteins. Genome segment sizes range from 600 to 3,000 base pairs for a total coding capacity of approximately 18,000 to 19,000 base pairs. Positive genome strands and messenger RNAs are capped but not polyadenylated. Negative strands have a diphosphate at their 5' end. There are short untranslated regions at the 5' and 5' and 3' ends of each genome segment. Replication is cytoplasmic but associated with discrete pseudo-organelles variously called virus factories, virus bodies, or viroplasm. After attachment to a receptor, the virion is brought into a cell by receptor-mediated endocytosis. In the endosome, cleavage of capsid proteins form the ISVP, or infectious subvirion particles. The ISVP is capable of breaching the endosomal membrane and gaining entry into the cytoplasm. Clathrin-mediated endocytosis is the predominant form of reovirus entry in nonpolarized cell lines, although alternative entry modes exist. The capacity to use multiple entry pathways might promote viral replication or pathogenesis in diverse cellular environments. ISVPs can also form in the intestinal lumen before entering a host cell, and they can then enter through endocytosis. On penetration of ISVPs into the cytoplasm, they are converted into cores by further loss of capsid proteins. This slide shows the changes that the virion goes through during the infectious cycle. What we see happening is that the virions are disassembled by acid-dependent proteases to form defined intermediates. So if you look at the slide, the figure at the top, that's the virion. That's what is infectious and enters the cell. In the next sequence, that's the ISVP. And you can see that some of the proteins have been stripped off from the virion. And then in the bottom illustration, that's the core. And only the inner proteins from that inner capsid remain. The others have all been stripped away. So the bottom is the core, where the double-stranded RNAs are going to be transcriptionally active. Each of the 10 segments of double-stranded RNA in the core is used to synthesize a messenger RNA molecule that is the same length as the plus strand of the genome. All of this primary RNA synthesis occurs within the core. Synthesis of the messenger RNA is conservative. The newly synthesized messenger RNAs are extruded from the core at the vertices, and both strands of the parental double-stranded RNA remain within the core. Of the proteins produced, 8 are components of the virion and 4 are non-structural, present only in the infected cell. The messenger RNAs also serve as intermediates in the replication of reoviruses. Following synthesis and release from the core, messenger RNAs quickly become associated with proteins to form complexes. These new cores form and double-stranded RNA is produced. Non-structural proteins are important for development of the viroplasm, an electron-dense area in the cell that contains a high concentration of viral proteins and messenger RNAs. Capsid assembly takes place in the viroplasm. In the model shown here, messenger RNAs are selected and packaged. RNA synthesis within the newly forming capsid generates the double-stranded genome segments. The outermost layer of the rotavirus triple-layered particle is obtained upon budding into the ER transport vesicles. So the viruses do not bud from the cell itself. They are dependent upon the cytoplasmic membrane breaking down so that they can be released. In terms of rotavirus disease, it's a diarrheal disease, causes several days of a very watery diarrhea. Sometimes there's nausea and vomiting along with it. You get mild fever, and so that's one way to recognize this in contrast to a bacterial gastroenteritis, because when a patient develops a bacterial enteritis, they usually have a high fever. And the virus is infecting columnar epithelial cells of the small intestine. The inner lumen of the small intestine is lined with columnar epithelial cells. And the surface, the apical surface of the columnar cells are covered with microvilli, because remember, these cells are important in absorption. And when these cells are infected with virus, you see shortening and blunting of the microvilli, and you see an infiltrate of the lamina propria, which is what keeps the individual cells lining the lumen of the gut stuck together, let's say. It's almost like cement, so that you don't get fluids leaking between the cells. Loss of fluids and electrolytes can be fatal. In the U.S., we have millions of cases each year. It results in a lot of visits to the doctor's office. Fortunately, not very many deaths. But that situation changes when we look in developing countries. There we see 600,000 to 1 million deaths per year, and that's just because these infants are much less likely to survive one of these infections than infants in the U.S. because of, issues such as lack of access to clean water, nutritional problems, and so on. And here you can see rotavirus is the most common cause of severe diarrhea amongst children, and it results in the hospitalization of approximately 55,000 children each year in the United States and the death of over 600,000 kids annually worldwide. The incubation period for the disease is only about 2 days. And again, the disease is characterized by particularly by diarrhea, and you get some fever, some abdominal pain. Immunity after infection is incomplete, but repeat infections tend to be less severe than the original infection. The primary mode of transmission is fecal-oral, although some have reported low titers of virus in respiratory tract secretions and other bodily fluids. Because the virus is stable in the environment, transmission can occur through ingestion of contaminated water or food in contact with contaminated surfaces. In the United States and other countries with a temperate climate, the disease has a winter seasonal pattern, with annual epidemics occurring from November to April. The highest rates of illness occur among infants and young children, and most children in the United States are infected by the time they reach 2 years of age. Adults can be infected, though the disease tends to be mild. For persons with a healthy immune system, rotavirus gastroenteritis is a self-limited illness and only lasts for a few days. Treatment is nonspecific and consists of oral rehydration therapy to prevent dehydration. About 1 in 40 kids with rotavirus gastroenteritis will require hospitalization for intravenous fluids. Okay, in this slide, you're looking at the causes of diarrheal diseases or gastroenteritis in children in developed versus developing countries. And you can see that the chunk that's attributable to rotavirus is the same, regardless of how well developed the country is or how primitive their resources might be in a particular country. So rotavirus is a problem in industrialized and developing countries. Infection will eventually confer natural immunity, but vaccines could be the best means of control. In 2009, the World Health Organization recommended that rotavirus vaccine be included in all national immunization programs. Rotavirus vaccines are licensed in more than 100 countries, but only 31 countries have introduced routine rotavirus vaccination. The incidence and severity of rotavirus infections has declined significantly in countries that have acted on the recommendation to introduce the rotavirus vaccine. For instance, in Mexico, which in 2006 was amongst the first countries in the world to introduce a rotavirus vaccine, the diarrheal disease death rates from rotavirus dropped by more than 65% among children aged 2 and under during the 2009 rotavirus season. In Nicaragua, which in 2006 became the first developing country to introduce the rotavirus vaccine, Investigators recorded a substantial impact, with rotavirus vaccine preventing 60% of cases against severe rotavirus and cutting emergency room visits in half. In the United States, vaccination has reduced rotavirus-related hospitalizations by as much as 86% since 2006. Each year, the vaccine prevents an estimated 40,000 to 50,000 hospitalizations among U.S. infants and young children. Rotavirus illness has also decreased among older children and adults that are not vaccinated. It's believed that vaccinated children provide indirect protection to other people because they're less likely to get and spread the disease. Currently, in the U.S., we have 2 vaccines that are licensed for use against rotavirus. We have Rotatech, which is a live oral pentavalent vaccine. So 5 different strains went into producing this vaccine. And then we have Rotatrix, which is also a live attenuated vaccine, but this one is monovalent. The vaccines are not without their controversies. First, there's a link between the vaccines and intussusception. Intussusception is the most common abdominal emergency in young children. What happens is that a segment of the intestine telescopes into itself, which is something that needs immediate medical attention. Rotavirus vaccine is the only medicine associated with the development of intussusception. However, the risk is low, about 1 to 6 excess cases of intussusception per 100,000 vaccinated infants has been documented. So, in general, it's been determined that slightly elevated risk of intussusception, which is something that can occur in children normally without any association with vaccine, it's been determined that that's an acceptable risk. Another issue was that DNA from porcine circovirus type 1 and type 2 was detected in the 2 vaccines used against rotaviral gastroenteritis. First found in Rotatrix, the FDA recommended a temporary suspension of the vaccine in the U.S. in March 2010 to further assess the issue. In May 2010, the FDA lifted this suspension, concluding that while live PCV1 was indeed detected in the vaccine, there was no evidence that the porcine circovirus type 1, or for that matter, circovirus type 2, caused any pathology in humans. So after that issue with the Rotatrix vaccine, they then found both PCV1 and PCV2 in Rotatech as well. But again, it's been determined that these porcine viruses do not pose a safety risk in humans, and they've not been shown to cause infection or illnesses in humans. One interesting result of vaccination with the rotaviral vaccines is that there's been shown to be a significant reduction in the incidence of type 1 diabetes in children who received the entire rotavirus vaccination series, compared to both a contemporary cohort and a historical cohort who were not vaccinated. There was no reduction in incidence in children who received only part of the recommended vaccination series. The pentavalent vaccine in particular was associated with lower risk. Rotavirus infection has been shown to accelerate the destruction of beta cells in diabetes-prone mice and leads to pathogenic infection of the pancreas. So the vaccines preventing the rotavirus infection may be indirectly protecting the pancreas from the damage from these ubiquitous viruses.